Hello everybody, I'm ARNAU, Yam geneticist at CIRAD. At CIRAD, in Guadeloupe and Martinique, we have implemented a participatory breeding approach. We create new progenies that we evaluate for several morphoagronomic quality-related traits during first 5 years. In a first phase, promising hybrids are evaluated on a larger scale in different environments, in partnership with the Agricultural Chamber and the Technical Institute. It takes about 10 years to develop a new variety. The traits evaluated at CIRAD are flesh color, what color is preferred, the presence of rods on tuber surface that make tuber unattractive and peeling difficult. We use a scale 0, 1, 2, 0 for absence, 1 some, and 2 a lot. The flesh browning, we measure the oxidation ratio, the length of oxidation 15 minutes after cutting the fresh tuber longitudinally. Measure from the head. The tuber shape measure has the length-width ratio. Oval and cylindrical, slightly elongated tubers are preferred. The regularity of shapes, because regular shapes are preferred, as they facilitate harvesting and storage, low risk of breaks and injuries, and also tuber peeling. This trait is strongly affected by environmental factors, particularly by soil structure, and it's why breeding for this trait is a long process. For flesh color, we have recently started to use image acquisition, as it allows to have information on color shades. In the phase 3, the hybrids are evaluated in different environments. Quality is evaluated by a hedonic test on boiled samples. We evaluate the acceptance of appearance, color, texture in the mouth, the taste, and global acceptance. Now I will briefly present CIRAD's activities in AfricaYam project, the phase 1. We conducted and meta-QTL analysis on 2D Alata biparental population generated between contrasting genitors. The objectives were the identification of genomic regions or markers associated with morphoagronomic characters that determine quality, the development of faster and more precise tools to improve the efficiency of yam breeding programs, and the understanding the genetic basis of quality traits. The major achievements are to deploy population segregating for K quality traits were generated, about 300 hybrids. Both populations were phenotypic for K quality traits. On the right, you can see the distributions of different traits for both populations, population A and population B. First correspond to tubercular roots, second to ratio length-width, first the shape regularity, and after the oxidation ratio, flesh color, and skin texture. The red arrow correspond to phenotypic values of female genitor and blue arrows to phenotypic values of males. Height variability was observed for each trait in both populations. Both populations were genotyping by sequencing, and a consensus genetic map was generated. We identified QTL for all traits. A total of 34 QTL were identified, explaining each between 11 and 43% of total phenotypic variance. QTL analysis provide also understanding of genetic basis of quality traits, quantitative versus qualitative, and genes mode of action. A total of 9 QTLs were validated on a diversity panel that will be useful for marker-assisted selection, both to select best parental combinations and promising hybrids in segregating population. On the right, you can see the distribution of phenotypic data in the validation panel according to the observed genotypic classes. Last figure corresponds to the alleles of marker validated for shape regularity. At this locus, heterozygote varieties AB present intermediate values than homozygote AA and BB. Has been a big work. The characterization of plants, the seed preparation, planting and phenotyping of 300 hybrids, 2 years to block, 9 replicates. A total of about 11,000 plants were analyzed. And all my team was involved in this work. And also Emmanuel ARNAU, PhD from Côte d'Ivoire, who came 2 times 6 months in Guadeloupe, financed by the RTBfoods project, as well as many students from Sherbrooke University of Canada. I thanks to different contributors from Serrat, Fabien Cormier, Denis Cornet, Eric Maledon, Eline Oudol, Hélène Ligne, Marie-Claire Gravillon, Pierre Mornay, and Anna Shahir. From CNRA of Côte d'Ivoire, the PhD Emmanuel ARNAU. From the Sherbrooke University, the students Emily Boulerne and Anne-Marie Lafontaine. Also our Caribbean partners, the Technical Institute and the Agriculture Chamber of Guadeloupe and Martinique. And I thanks also the following projects that make the research possible: the AfricaYam project and the RITA project of enlargement of the yam variety portfolio. Thanks for listening.